Hello students, today we are going to discuss a very important topic known as the nucleus. This nucleus was first of all discovered by a scientist whose name was Robert Brown from the cells of the orchid plant. And also there was a scientist whose name was Strasburger, who gave the statement that each nuclei arises from the pre-existing nuclei. And we must also know that nucleus is the control center of the cell. We can say that it is the controller of the cell, it is director of the cell. And this statement that it is the control center of the cell was first of all given by a scientist known as the Hammerling. Hammerling did his experiments on a unicellular algae known as the Acetabularia and came to the conclusion that nucleus is the control center of the cell. But you have to remember this thing, that it's not necessary that a well-defined nucleus is present in all the cells. In the previous videos also I have explained you that there are certain cells known as the prokaryotic cells in which the nucleus is not well defined and it is not bounded by a well-defined nuclear membrane, and such a nucleus is called as incipient nucleus, or we can say that nucleoid. Means what I want to say, in prokaryotic cells a well-defined nucleus is absent and they have an incipient nucleus which is also called as the nucleoid. But the presence of the well-defined nucleus is the feature of the eukaryotic cells. But there are few exceptions in eukaryotes also where the nucleus is absent in the cells. For example, in the mammal RBC, and the sieve tubes of the phloem, mammal RBC and the sieve tubes of the phloem are the two examples where the nucleus is absent. But in few mammals such as the camel and the llama, the nucleus is present. Okay, now before discussing the details of the nucleus, you must know that what is the chief function of the nucleus. As we know very well that nucleus consists of chromosomes, it consists of DNA, it consists of genes. So I can say that it is a storehouse of the genetic material. Not only this, when we study the cell division, the top priority is that the nucleus first divides and then the cytoplasm divides. We know very well karyokinesis is followed by cytokinesis. It means nucleus plays a very important role in the cell division also, and it is helpful in the transmission of the genetic information. The genetic information from one generation to another generation is actually moving through the nucleus only. Okay, now when we have to discuss the nucleus in detail, we must study some important points such as the number of the nucleus, the shape of the nucleus, the size of the nucleus, the chemical composition of the nucleus, and the structure of the nucleus. So very first we are discussing about the nucleus. The first point, that is, what is the size of the nucleus? The first point is the size of the nucleus. Size of the nucleus varies from 5 micron to 30 micron. Okay, this is the size of the nucleus. If we talk about the shape of the nucleus, then shape of the nucleus varies. It may be spherical, it may be oval, it may be elongated, etc. It has various shapes, but I will be quoting here an example of leukocytes. What are leukocytes? Leukocytes are basically WBC. Why I have quoted this example here? Because in leukocytes the shape of nucleus is irregular. They are not having a definite shape. Okay, otherwise the nucleus may be spherical, oval, elongated, etc. But in leukocytes the shape of the nucleus is irregular. So this size This is shape. Now coming to the number of nucleus. Now there are three conditions. The first condition is mononucleate condition. What do you mean by this mononucleate condition? Mononucleate condition means one nucleus per cell. Generally, mononucleate condition is found in maximum cell. The number of the nucleus per cell is 1 in number. When there is one nucleus per cell, then it is mononucleate condition. It's very common condition found in the various cells. Number 2 condition is known as the binucleate condition. Now what is binucleate? Bi means 2. So 2 nucleus per cell. And this binucleate condition is present in the paramecium. If we see the structure of the paramecium, then you will be able to see that inside the paramecium there are 2 nuclei. One is known as the macronuclei and another is known as the micronuclei. So we can say that there are 2 nuclei. One is small, one is big. Small one is known as micronuclei, the large one is known as what? The macronuclei. So there are 2 nuclei, we can say it is binucleate condition. But the very important thing is the polynucleate condition. What is polynucleate condition? Polynucleate condition means many nuclei per cell. Now the question raises that how this polynucleate condition is generated, how it is produced. So the polynucleated condition may be of 2 types. Okay, what I am saying, that polynucleate condition may be of how many kinds? 2 kinds. One is known as the syncytium. Now what is syncytium? Syncytium condition is formed by fusion of cells. It is produced by, or it is formed by the fusion of the cell. Syncytium means what? Here syncytium means what? Multinucleate condition only, right? But how the multinucleate condition is formed? So many cells are fused together and they form a syncytium, that is the multinucleate condition. And the second condition is known as cenocyte. It is produced because of free nuclear division. Here also multinucleate condition is produced. Here also multinucleate condition is produced. But what is the chief difference? That if the multinucleate condition is formed by the fusion of the cell, it will be called as the syncytium. And if the multinucleate condition is formed by the free nuclear divisions, then the condition will be called as what? Cenocyte. So both syncytium and the cenocyte produces what? The polynucleate condition. So students, this was all about the number of the nuclei, that it may be 1 mononucleate, 2 binucleate, many means what? Polynucleate. So this was about the size, shape, number. Now have a look on chemical composition. That what is the chemical composition of the nucleus? Nucleus is composed of basically protein. It is also composed of phospholipids, okay, DNA and also RNA. Okay, what is the chemical composition of the nucleus? It consists of proteins, phospholipids, DNA and RNA. So just now we have discussed about the size, shape, number and chemical composition. But the very important thing is the structure of the nucleus, right? We are going to discuss the structure of the nucleus. Structure of nucleus. To study the structure of the nucleus, we have to discuss 4 points. Okay, the very first is known as the karyotheca. Karyotheca can also be called it as nuclear envelope, or it can also be called it as nuclear membrane. Okay, you can see I have made the nuclear membrane. This diagram is suggesting the structure of the nucleus. Okay, so this is what the nuclear membrane. This is the nuclear membrane. Actually, nuclear membrane is separating the nucleoplasm and the cytoplasm. Okay, here what is filled inside the nucleus? What is filled? Nucleoplasm. Outside the nucleus, what is present? Cytoplasm. So the cytoplasm and the nucleoplasm are well separated by the help of what? The karyotheca, or the nuclear envelope, or the nuclear membrane. The second one is the fluid filled inside. That is known as what? Nucleoplasm. Nucleoplasm, it is also called as karyoplasm. The fluid which is filled, inside the nucleus is called as what? The karyoplasm. The third one is known as what? This one, that is known as the, two things are there. One is known as the nucleolus, and the fourth one is known as the chromatin net, or we can say that chromatin threads. Okay, so there are how many components of the nucleus? There are 4 components of the nucleus: karyotheca, nuclear envelope, or the nuclear membrane. The second is the nucleoplasm. The third one is known as the nucleolus, and the fourth one is the chromatin net, which are actually nothing. They will form the chromosomes. They will form the chromosomes at the time of what? The cell division. Okay, so let's have a discussion on the first point, first component of the nucleus, known as the karyotheca. Karyotheca is basically what? The nuclear membrane. Always remember that nuclear membrane is double membrane. It's not single membrane. There are 2 membranes. A nucleus is surrounded by 2 membranes. One is known as what? The outer one, and the second one is known as the inner one. Okay, it is also 75 angstrom in thickness. It is also 75 angstrom in thickness, and in between these two membranes, perinuclear space present. Are you able to see? This is outer membrane, this is inner membrane, and in between the outer and the inner membrane, what is present? Perinuclear space is present, and it is of what? 15 nm. This space is having the size of 15 nm. Okay, so in between the two membranes, what is present? Perinuclear space is present. Now outer membrane is studded by ribosomes, and it is in continuation with ER. You can see here that on the outer membrane, what are present? Ribosomes are present. These black dots what I have made are nothing, they are the ribosomes, and this outer membrane is in continuation with what? The ER. Okay, now one thing what you are seeing, that the nuclear membrane is not continuous. The nuclear membrane is not continuous. Nuclear membrane is not continuous, and it has several pores, right? Several pores called what? Nuclear pores. Nuclear pores of size 10 nm to 100 nm. Okay, so the nuclear membrane is not continuous, and it has pores at certain places, and these pores are called as what? Nuclear pores, and the size of the nuclear pore is 10 nm to 100 nm. Okay, now through these pores, ribosomes can pass. Through these pores, what can pass? Ribosomes can pass, having the size of 230 to 250 angstrom. Ribosomes are having the size of 230 to 250 angstrom, which can pass through these nuclear pores. Okay, now what happens? That these nuclear pores are having certain plugs made of nucleoplasmin protein, called annulus, which is also called as blab, called as annulus. Remember, that is pores, nuclear pore plus annulus is equal to pore complex. Are you getting? First, what I have said, that nuclear pores are present in the nuclear membrane, but these nuclear membranes, these nuclear pores are having certain plugs which are made up of nucleoplasmin protein, and these plugs are called as what? Annulus. Nuclear pore plus annulus forms what? Pore complex. Now the question arises that what is the role of this pore complex? What is the role of this pore complex? So remember that pore complex avoid free nucleocytoplasmic exchange. Okay, means the movement between the nucleoplasm and the cytoplasm is not free. Only selective substances will be passed from nucleus or through the cytoplasm. Okay, so the pore complex avoid free nucleocytoplasmic exchange. Okay, now just below nuclear membrane, is present a protein network, filamentous protein network called nuclear lamina. Okay, what is the function? What is the function of this nuclear lamina? It helps in growth of nuclear envelope, and it also helps in attachment of chromosomes. It helps in attachment of chromosomes. Okay, so this was about the first component of the nucleus, and that is known as the nuclear membrane. Now coming to the second one. What is the second one? That is nucleoplasm. This nucleoplasm term was given by Strasburger. Okay, and this is transparent granular substance filled in nucleus. And now what it consists of? So it consists of nucleotides, it consists of nucleosides, it consists of various enzymes such as DNA polymerase, RNA polymerase. Okay, it consists of proteins, minerals, it is magnesium ions, calcium ions, etc. It consists of the nucleotides, nucleosides, enzymes, proteins, minerals, etc. So this is the composition of what? The nucleoplasm. And what the thing you have to note here is that the other two components, the rest remaining two components known as the nucleolus and the chromatin threads, nucleolus and the chromatin threads are present in nucleoplasm. Are you getting? The rest two components, that is known as the nucleolus and the chromatin thread. Nucleolus and the chromatin threads are present in what? The nucleoplasm. Okay, so now coming to the third component, that is the third one is what? The nucleolus. Nucleolus, it was discovered by Fontana, scientist name Fontana, name given by given, the name Nucleolus was given by the Bowman. Okay, Nucleolus is membrane-less. Nucleolus consists of DNA, RNA and protein. It consists of nucleolus, RNA and protein. One nucleus has one nucleolus, but more than one nucleolus may be present. For example, in onion, 4 nucleoli present. Okay, and what is the location of the nucleoli? What is the location of the nucleolus? So it is present at Nucleolus organizer region. Earlier also in my videos, in the video that was the named chromosome, I have explained the Nucleolus organizer region. In chromosome there is a region known as the Nucleolus organizer region. Why it is called as Nucleolus organizer region? Because Nucleolus organizer region of the chromosome consists of what? The nucleolus. So we can say that what is the location of the nucleolus? It is present at the Nucleolus organizer region. Okay, now a nucleolus consists of 3 parts. A nucleolus consists of 3 parts. Okay, first part is known as fibrillar part. Fibrillar part, it consists of fibrils made of ribonucleoprotein, right? Second part is granular part. This part consists of granules made of ribonucleoprotein, and the third part is known as pars amorpha, and this is the matrix part. This is the matrix part in which fibrils and granules present. Okay, what I have said, these fibrils and the granules both are present in the pars amorpha, and the chief function of the nucleolus is rRNA synthesis. Means we can say that ribosomes are synthesized here. As we know very well कि ribosomes are made of rRNA, so we can also say that ribosomes are synthesized here. So this is the chief function of the nucleolus. And nucleolus, where the nucleolus is present? So the nucleolus is present in the nucleoplasm. The fourth part is also present in the nucleoplasm only, and that is known as what? The chromatin net. This term chromatin net was given by Fleming, right? And these are thread. This chromatin net is made of threads which can be stained by basic dyes, right? Or we can say that basic stains, right? So the chromatin net are actually made up of the threads which can be stained by the basic stains, and this chromatin net term was given by Fleming. This term was given by what? The Fleming. And this chromatin net at time of cell division become condensed to form what? The chromosomes. And we know very well that chromosomes are hereditary vehicles which carry the genes from one generation to another generation. So at the time of the cell division, this chromatin net become condensed and form what? The chromosomes. Okay. And earlier also in my videos I have said about, you can go in details in my previous videos also, right, that chromatin is of two types. One is known as euchromatin and the another one is known as heterochromatin. Okay, and this is darkly stained part, this is darkly stained part, and this is lightly stained part. Okay, it is genetically active and it is genetically non-active part. It helps in transcription and it is not participating in transcription. Okay, so these were the four components of the nucleus. If you have liked this video, subscribe my channel, also like and share this video. Thanks a lot.